Since the beginning of life on Earth, there has been exploration to know the unknown. Charts and maps based on the data are designed from time to time, which enable the subsequent explorers to push ever farther the boundaries of their knowledge about the earth, the sea, the sky, and indeed ourselves. These researchers even reached the human cell, which helped the scientists to reach to a stage where they could explore biology's most important mapping expedition, the Human Genome Project.
Hello students, I welcome you all to today's lesson. ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਜਾਣਦੇ ਹੀ ਹੋ ਕਿ ਸਾਡੇ ਸਰੀਰ ਵਿੱਚ ਸੈੱਲ ਮੁੱਖ ਭੂਮਿਕਾ ਨਿਭਾਉਂਦੇ ਹਨ। ਇਹ ਖਿਆਲ ਰੱਖਦੇ ਹਨ ਕਿ ਕਿਸ ਤਰ੍ਹਾਂ ਵਧਣਾ ਹੈ ਅਤੇ ਕਿਸ ਤਰ੍ਹਾਂ ਕੰਮ ਕਰਨਾ ਹੈ। ਇਨ੍ਹਾਂ ਸਾਰੀ ਕਿਰਿਆਵਾਂ ਲਈ ਕੁਝ ਤਰਤੀਬਬੱਧ ਸੂਚਨਾ ਸਾਡੇ ਸੈੱਲਾਂ ਵਿੱਚ ਜੀਨਸ ਦੇ ਰੂਪ ਵਿੱਚ ਪ੍ਰੈਜ਼ੈਂਟ ਹੁੰਦੀ ਹੈ। ਜੀਨਸ क्रोमोसोम्स ਦੇ ਉੱਤੇ ਲੰਮੇ ਪੋਲੀਮਰਸ ਦੀ ਫਾਰਮ ਵਿੱਚ ਮੌਜੂਦ ਹੁੰਦੇ ਹਨ। We have 46 chromosomes arranged in pairs and packed in the nucleus of the cells. The chromosomes are made up of deoxyribonucleic acid or DNA and proteins. Components of DNA like sugar, phosphate, and a nitrogenous base jointly become the building blocks for our genes. The genes string together to make our chromosomes. We have heard the term gene many a times, but do you know about the human genome?
Genome is basically the total genetic material contained within the chromosomes of an organism. Means all genes together known as genome. A genome is a complete set of coded instructions for making and maintaining an organism. The term genome was coined by H. Winkler in 1920. There is another related term, genomics. It is the scientific discipline of mapping, sequencing, and analyzing genome. The term genomics was coined by Thomas Rodrigues in 1986. Actually, when we use this term genome, we mean complete DNA in an organism. DNA, along with other components, is made up of 4 nitrogenous bases that are abbreviated as A, T, C, and G. These bases are repeated millions or billions of times throughout a genome. What is important about these bases is the order in which these bases are arranged. This order dictates whether an organism is human or another species, such as yeast, rice, or fruit fly, because all organisms are related to each other through some similarities in their DNA sequences. So, knowledge gained from other creatures' genome always help to know about human biology. Today, we will learn about the concept human genome project.
ਤੁਹਾਨੂੰ ਇਹ ਜਾਣ ਕੇ ਹੈਰਾਨੀ ਹੋਵੇਗੀ ਕਿ ਹਿਊਮਨ ਜੀਨੋਮ ਪ੍ਰੋਜੈਕਟ ਭਾਵ HGP ਇਤਿਹਾਸ ਵਿੱਚ ਇੱਕ ਅਦਭੁਤ ਖੋਜ ਹੈ। ਇਸ ਅੰਤਰਰਾਸ਼ਟਰੀ ਖੋਜ ਦੇ ਕਾਰਨ ਹੀ ਮਨੁੱਖ ਦੇ ਜੀਨਸ ਦੀ ਸੀਕੁਐਂਸ ਅਤੇ ਮੈਪਿੰਗ ਸੰਭਵ ਹੋਈ। The Human Genome Project as mega project was a 13-year-long project that formally began in October 1990 and was completed in 2003. Originally, the project was planned to last 15 years, but rapid technological advances accelerated the completion date to 2003. The US Department of Energy and the National Human Genome Research Institute, that is NHGRI, together sponsored this project. It aimed to discover the estimated 20,000 to 25,000 human genes and make them available for further biological study. The HGP gave us the ability to read nature's complete genetic blueprint for building a human being. In our today's class, we will discuss some important aspects of this mega project like goals of Human Genome Project, salient features of Human Genome Project, applications of Human Genome Project, future challenges of Human Genome Project. Let us start with goals of Human Genome Project.
Goals of Human Genome Project: to identify all the genes in human DNA, which are approximately 20,000 to 25,000 in number, also to determine the sequences of the 3 billion chemical base pairs that make up human DNA. Human Genome Project was designed to determine the functions of all the genes and to identify various genes that cause genetic disorders. After getting all this information, HGP also planned to store this information in databases to provide proper tools for analysis of this data, along with an objective of transferring related technologies to the private sector while dealing with the making of mankind. Human Genome Project also took the responsibilities to address the ethical, legal, and social issues that may arise from the project.
ਵਿਦਿਆਰਥੀਓ, ਇਨ੍ਹਾਂ ਗੋਲਸ ਨੂੰ ਪੂਰਾ ਕਰਨ ਲਈ ਵਿਗਿਆਨੀਆਂ ਨੇ ਵੱਖ-ਵੱਖ ਗੈਰ ਮਨੁੱਖੀ ਸਜੀਵਾਂ ਦੇ ਜੈਨੇਟਿਕ ਮੇਕਅਪ ਦਾ ਵੀ ਅਧਿਐਨ ਕੀਤਾ। ਇਨ੍ਹਾਂ ਵਿੱਚ ਪ੍ਰਮੁੱਖ ਸਨ ਹਿਊਮਨ ਗਟ ਵਿੱਚ ਪਾਇਆ ਜਾਣ ਵਾਲਾ ਬੈਕਟੀਰੀਅਮ ਐਸਚਰਚੀਆ ਕੋਲਾਈ, ਫਰੂਟ ਫਲਾਈ ਅਤੇ ਲੈਬੋਰੇਟਰੀ ਮਾਊਸ। A unique aspect of the Human Genome Project is that it was the first large scientific undertaking to address potential ethical, legal, and social issues arising from project data. Another important feature of the project was the federal government's long-standing decision to transfer the technology to private sector. By licensing technologies to private companies and by awarding grants for innovative research, the project helped US biotechnology industry and to come out with some new development in medical field. But the real question here is, why was the Human Genome Project initiated?
Reasons to start Human Genome Project. Major concern for starting such a huge project was to have some control on hereditary diseases. You know, there are more than 3,000 disorders known to result from a single altered gene, and these diseases affect millions of healthy lives. After having complete knowledge of the involved gene, scientists could do genetic alterations to avoid a particular disease. For example, progress in understanding the causes of cancer could be possible only after the recent discovery of cancer genes. The goal of the Human Genome Project is to provide scientists with powerful tools and new information to help them clear the research hurdles. Causes and actions against many tragic and devastating illnesses such as schizophrenia, alcoholism, Alzheimer's disease, etc., are also being studied, probably due to the knowledge of human genome. Finally, Human Genome Project research would help to solve one of the greatest mysteries of life. How does one fertilized egg give rise to the different specialized cells, such as those making up muscles, brain, heart, eyes, skin, blood, and so on? For a human being or any organism to develop normal, a specific gene or sets of genes must be switched on in the right place at the right moment during the development process. Information generated by the Human Genome Project will help to control all those activities happening at gene level which may go wrong during fetus developmental process.
ਅੱਗੇ ਵਧਣ ਤੋਂ ਪਹਿਲਾਂ ਚਲੋ ਕੁਝ ਡੈਫੀਨੇਸ਼ਨਸ ਨੂੰ ਡਿਸਕਸ ਕਰ ਲਈਏ ਜੋ ਕਿ ਹਿਊਮਨ ਜੀਨੋਮ ਪ੍ਰੋਜੈਕਟ ਨੂੰ ਸਮਝਣ ਲਈ ਬਹੁਤ ਜ਼ਰੂਰੀ ਹਨ। cDNA ਮਤਲਬ complementary DNA. Complementary DNA was prepared by cloning the DNA generated from mRNA synthetically. These were stored in the form of library. These libraries are very important to scientists as they contain clones of all protein including DNA. MB matlab megabase. It is unit of length equal to 1 million base pairs. Microarray. It is a technique used to identify and classify DNA sequencing information. A microarray works by exploiting the ability of a given mRNA molecule to bind specifically to the DNA template from which it originated. They can be used to study how large number of genes are expressed as messenger RNA. एक और term है STS which stands for sequence tagged site. It is a short DNA segment usually of 200 to 500 base pairs. It occurs only once in a genome and its location and order of bases is known. STSs serve as landmarks on the physical map of a genome. चलो हुन कुछ techniques and methodologies ਦੀ ਗੱਲ ਕਰੀਏ जो कि ह्यूमन जीनोम प्रोजेक्ट विच यूज ਕੀਤੀ ਗਈ।
Techniques and methodologies. The methods involved two major approaches. Expressed sequence tags, that is EST. This approach was used to identify all the genes that are expressed as RNA. Sequence annotation. This approach was used to sequence the whole set of genome that contains all the coding and non-coding sequence, and later identifying different regions in the sequence according to their functions. Another techniques which were used in the HGP were RFLP, which means restriction fragment length polymorphism, YAC, yeast artificial chromosomes, BAC, bacterial artificial chromosomes, PCR, polymerase chain reaction, electrophoresis, etc. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Here, you remember that DNA is a very long polymer. It is cloned in suitable host using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called as BAC, bacterial artificial chromosomes, and YAC, yeast artificial chromosomes. The fragments were sequenced using automated DNA sequences. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was not possible otherwise. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed in May 2006. This was the last of 24 human chromosomes. Another challenging task was assigning the genetic and physical maps of the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites.
ਹਿਊਮਨ ਜੀਨੋਮ ਪ੍ਰੋਜੈਕਟ ਮਨੁੱਖ ਦੀ ਬਹੁਤ ਹੀ ਮਹੱਤਵਪੂਰਨ ਤਰਤੀਬਬੱਧ ਵਿਗਿਆਨਿਕ ਕੋਸ਼ਿਸ਼ ਹੈ। ਇਸ ਪ੍ਰੋਜੈਕਟ ਵਿੱਚ ਸਿਰਫ ਜੈਨੇਟਿਕਸ ਨਾਲ ਸੰਬੰਧਿਤ ਲੋਕ ਹੀ ਸ਼ਾਮਲ ਨਹੀਂ ਸਨ, ਬਲਕਿ ਹਰ ਖੇਤਰਾਂ ਦੇ ਲੋਕ ਜਿਵੇਂ ਭੌਤਿਕ ਵਿਗਿਆਨੀ, ਰਸਾਇਣ ਵਿਗਿਆਨੀਆਂ ਤੋਂ ਲੈ ਕੇ ਇੰਜੀਨੀਅਰਸ ਅਤੇ ਰੋਬੋਟਿਕਸ ਦੇ ਨਾਲ ਕੰਪਿਊਟਰ ਇੰਜੀਨੀਅਰਸ ਆਦਿ ਨੇ ਵੀ ਮਹੱਤਵਪੂਰਨ ਯੋਗਦਾਨ ਪਾਇਆ। Never before in the history of humanity had so many professionals united for a single scientific endeavor. Students, can you imagine the real cost of this project? The total cost of the Human Genome Project is unbelievable. You know, about $3 billion was spent on this project. Why did it cost so much? Well, consider all of the people and equipments involved. Computers were needed to store the data. Technicians were needed to maintain the computers. Geneticists were engaged to map the genome. Most modern laboratory equipments were required. So much effort in terms of men and machines were involved. But for what? Of course, all for the welfare of mankind. Identifying 3 billion letters which spell out the DNA in our human genome blueprint that makes us human was an awesome accomplishment. Some have compared it to the development of the periodic table. Others have compared it with the invention of the wheel. Although the technical use of the Human Genome Project information to cure disease is extremely significant, drawing correct conclusions about our origin is also a major aspect of this project. Let us try to list findings of this project.
Key findings of Genome Project. Human genome has 3.1647 billion nucleotide base pairs. There are approximately 30,000 genes in human beings, the same range as in mice and twice that of roundworms. All human races are 99.99% alike, so racial differences are genetically insignificant. Only 0.1% of human genome, with about 3.2 million nucleotides, represents the variability observed in human beings. The functions of over 50% discovered genes are unknown. Chromosome 1 has 2,968 genes, while Y chromosome has 231 genes. These are the maximum and minimum genes for human chromosomes.
At about 1.4 million locations occur single nucleotide differences called SNPs or single nucleotide polymorphism. They have the potential to find chromosomal locations for disease-associated sequences and tracing human history. Repetitive sequences, which are repeated 100 to 1,000 times, make up large portion of human genome. They have no direct coding function but provide information regarding chromosome structure, dynamics, and evolution. Approximately 1 million copies of short 5.8 base pair repeated sequence are clustered around centromere and near the ends of chromosome. These represent junk DNA. This Human Genome Project was a mega project because if the sequence obtained were to be stored in typed form in a book, and if each page contained 1,000 letters and each book contains 1,000 pages, then 3,300 such books would be needed to store the complete information. The enormous quantity of data expected to be generated also became the necessity of having high-speed computer, large-sized hard drives for data storage and supercomputers for retrieval and analysis. As the project was a mega project, its application was also very large. diseases are more easily diagnosed. Sometimes, even before symptoms appear, doctors can identify the patients at risk simply by examining their genes. Gene therapy correcting diseases via genetic engineering can also help in curing more diseases as more disease-causing genes have been located. Let us quickly look at its extensive applications.
Applications of Human Genome Project HGP. Diagnosis of disorders. Geneticists cloned genes responsible for genetic disorders. In recent years, many achievements have been marked, like the cloning of genes responsible for Duchenne muscular dystrophy (DMD), retinoblastoma, cystic fibrosis, and neurofibromatosis. If other disease-related genes are isolated, biologists will be able to learn about the structure of the genes and their corresponding protein. This knowledge could help pathologists to diagnose and manage the resulting disorders. Prediction of disorders. Genome information can indicate the future likelihood of some diseases. For example, if the gene responsible for cancer disease is present, it is almost certain that symptoms would eventually occur. Genome information also helps geneticists to predict the individuals having an increased susceptibility to disorders such as heart disease, cancer, or diabetes. Healthcare. It will be helpful in healthier living, designing drugs and genetically modified diets. Insight into basic biology. Information generated by HGP may answer to several interesting biological questions. The discovery of new genes, along with their functions, is an obvious benefit. When biologists compare the human genome with the genomes of other organisms, they gain some insight into molecular evolution, including human evolution. Non-human organisms. Information about natural capabilities of non-human organisms can be used in meeting challenges in healthcare, agriculture, energy production, and some environmental problems. For this, a number of organisms have been sequenced, such as bacteria, yeast, etc. Development of new technologies. The HGP has catalyzed enormous advances in the development of technology, and it will continue to do so. Due to HGP, researchers have made great progress in developing automated machines and robotic workstations to perform repetitive procedures.
One of the biggest challenges of the HGP is finding faster ways to sequence DNA. Determination of the entire DNA sequence contained in the human genome will not answer all the questions. Applications of these outcomes will help the human race. So we can conclude that the Human Genome Project is a phenomenal undertaking. Unfortunately, it reminds us that some of the worst events in human history have occurred when technological expertise was united with spiritual emptiness. So students, enough for today. Let us quickly check with some short questions what you have grabbed today. Be attentive. Here are some questions. First question. Define a genome. Answer. A genome is a complete set of coded instructions for making and maintaining an organism. Next question. What is the Human Genome Project? The answer is, the Human Genome Project was a 13-year international project that aimed to determine the complete sequence of nearly 3 billion DNA subunits or bases, identify all human genes, and make them accessible for further biological study. Next question. What are the potential benefits of human genome research? Answer. The project brings in fantastic benefits for mankind. Generations of biologists and researchers have been provided with detailed DNA information that can be used to understand the structure, organization, and function of genes on the chromosomes. It will help in prediction and diagnosis of disorders to a large extent. Next question. Why is genome important? Answer. A genome refers to entire DNA in an organism. Genes carry information for making all the proteins required by the organism. Along with other things, these proteins determine how the organism looks, how well its body metabolizes food or fights infection, and sometimes even how it behaves. ਬੱਚਿਓ, ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਹਾਨੂੰ ਹਿਊਮਨ ਜੀਨੋਮ ਪ੍ਰੋਜੈਕਟ ਦੇ ਬਾਰੇ ਸਭ ਕੁਝ ਸਮਝ ਆ ਗਿਆ ਹੈ. Thanks for your cooperation and attention. Looking forward to the next class. See you then. Thanks.